on this part, but you know, such is life. So one year, my students, it was my birthday, and they were like, oh, like they knew I was all like about the coffee, all about that coffee life. And they were so sweet. In one year, I had 4 different students like bring me a Starbucks coffee, and I was like, oh my God, this is the sweetest thing like ever. And of course, like you can't like refuse a gift. That's just rude. That's like poor form. So what did I do? Oh, I drank those coffees, and they were all like grande coffees. And so like by 5th period, like 4th period, I was like next level, like in orbit. I was like, okay, guys, we're gonna do this. We're gonna do this. We can do this. And they were like, what is happening? He was like, I've had so much coffee. This is great. We can do this. Let's go run a mile. Let's do it. Like, yes. And they were like, oh my gosh. And then the drive home, because you climb, you know, it's the caffeine, like it's a stimulant. And then the higher you climb, the harder you fall. So the drive home, I was like, oh, I'm gonna barf. I can't do this. Oh my God, I'm gonna die. I was like, I can't. I legit had a headache all that night. Like I had to like it was a Friday. I had to put myself to bed. I was like, I can't handle this. I can't do it. But I was like, it was so nice. My husband's like, why did you do that? Like that's way too much. And I'm super sensitive to like medicines and caffeine and all that, like stimulants and stuff. Oh man. He was like, that was not smart. And I was like, I know. It was so good. It was so sweet and it was kind and I loved every minute of it. And then wasn't into the crash wasn't so great. Oh, I was vibrating, not just shaking, full on. Like I was steadying my hand and I was like I had somebody like grab my hand and they're like, oh my gosh, you are like tremors. It was not good. I'm pretty sure I felt like almost my heart stopping. Don't do that. Don't do that. Yeah, Brock.
Okay. Yeah, it's not the coffee that hurts you or the, afterwards. It's the caffeine. And I'm only knowing all this because I'm in a, health class. Yeah.
Yes. Oh yeah. Yep. Caffeine's a drug. I know it. We all know it. I'll just go to Caffeine Anonymous, Coffee Anonymous.
Yeah, I used to do caffeine.
CA.
I used to do a lot of sugar. It's not good for me. I go really high, then really low. I get really grumpy, and I don't like being grumpy.
Yes, and who does? All right, guys. Well, I know we'll have to do some polls later and be like, who is Team Coffee? Who is not Team Coffee? But let us get going because we got some other stuff we unfortunately or fortunately, depending on how you're feeling about this unit, get to discuss. Make sure my screen's doing well here. We are going to look at kind of recapping the second part of our notes that we did at the end of last week and I didn't even add it, I should add it right here, the elaborate section of your notebook, but we're going to do it together. So that's going to kind of be nice. Everybody should get all of their points on that one. Elaborate. Modeling. Okay, so we're going to do these 3 things today. So we're going to do a quick recap of the notes. We're then going to do the elaborate section of your notebook, which is slide 16. There's like 4 questions on it. We're just going to do it all together. That way kind of checks it off the list. And then you guys are going to, that will serve as your review, and that will allow you guys to take the second part of your mid-unit quiz, which is about the sources of genetic variation. Basically, all of the things we're going to talk about, this is going to serve as kind of like the big review before you guys do that. Okay, so this will allow you guys to take questions or ask questions, get everything clarified before we do that. And then tomorrow we'll be moving on to part 2 of the unit, where we get to start looking at like probability and like figuring out, okay, well, what are the chances of inheriting certain things, or what are the chances of certain combinations of traits showing up in an offspring, which is more of the fun part about it. Okay. So your big learning target states that you should be able to model all 3 of the different ways that we would be able to increase genetic variation through sexual reproduction. So that would be like recombination or crossing over. It would be what we call independent assortment that you guys looked at, and fertilization. So can you demonstrate like your understanding of those three concepts? All right. Now, first thing though, let's go back over because yesterday, and before I move on, I did post the video for yesterday's stuff. So if you were not able to make it yesterday for whatever reason, I know Mondays are a little nuts. We sometimes forget like the time change in the class. I posted that video as well as the document that I was kind of going over to the explain section, as well as to the daily class recording for yesterday's class. So it's in two different places for you. So you guys have that. But yesterday we looked at homologous chromosomes as a way of how our body stores and organizes its genetic information. Okay, remember each chromosome comes in a pair. Okay, so when we talk about you have 46 chromosomes, yes, that is absolutely true for humans. However, they are organized into sets. So we have 46 total, but we really have 23 pairs. And that's where we have these guys, the homologous chromosomes, or a lot of times you'll hear them be referred to as homologous pairs. And this is basically the same thing, okay? They're just saying these are homologous pairs. And when we use the term homologous, remember that prefix homo in science means same. So they're gonna have the same types of traits or genes in the same locations for each of those chromosomes, okay? And in that set, the reason you have a set is because you are going to inherit one copy of the homologous chromosome pair from your mom and one copy from your dad, okay? Now, the whole purpose of sexual reproduction, right, is to create these genetically unique offspring where you're going to donate half of your DNA. So basically, half of your total set that you have in your body right now, half of that would then go to your offspring. So remember we said that your DNA is kind of like a set of encyclopedias where you have all of these volumes. You have 23 pairs. That one chromosome from each one of those 23 pairs will end up going to your offspring later on in life, okay? Meiosis is the process of taking and putting half of your genetic information into either a sperm or an egg. It's how we cut that chromosome number in half to make your sex cells. So that when we go through sexual reproduction, the offspring has one half here, one half there, and one half plus one half equals one whole set, okay? Now, the cool thing, or what really has to take place first, and this is where, Carson, you were asking the other day about sister chromatids. This is where this comes in right now. When you, right before, the first thing that actually has to happen before any process of making a new cell. So in this case, we're making sperm and egg cells. We're making sex cells. And sex cells are called gametes. It's like just the formal scientific name for your sex cells. So if you see the word gametes, it just means your sperms or your eggs. Now, what has to take place first in order to maintain the correct amount of chromosomes and to ensure that the correct amount of chromosomes goes to each sex cell you're able to produce? They have to make a copy of themselves first. So each chromosome in the pair, so this would be your homologous pair, is going to make a copy of itself. Okay, so this copy from, we'll say the greens are from moms and the purples are from dads. So this green copy of this homologous pair is going to make a copy of itself. And that's where you get that X shape. This purple copy that you inherited from dad that makes up this set, this first set of chromosomes, gets copied and now looks like that traditional X shape that some people see. Now, the reason why they look like this X shape is because it's how they're connected. Right here, see how there almost looks like a little button or like a little, I kind of like to think of it because we call this the waist or the centromere of the chromosome. It's the central part. This is kind of like the belt buckle, okay? Because they're kind of connected at the hip and you wear your belts at your hip. And so it's kind of like this little like belt buckle spot that's going to keep the original copy and its clone. Okay. Now, when they're in this formation where you have what the original and the identical copy, we refer to them now as sisters. Like think like identical sisters or Siamese twins. Okay, because they are exactly identical copies. This is when you refer to them as sister chromatids. Again, I'm not going to test you in this part. So if you're like, that's a lot of vocab and different ways to talk about the chromosomes, you're right. It is. It's super annoying. Every year we're all like, oh God, well, who decides to call them all these different things? Okay, but just know when they're in this formation, when you see them formed as an X, it means that that cell, if we were to look at them under a microscope, which we're going to look at in just a second, this would mean that this cell is getting ready to create sex cells. It's ready to go through division and it's ready to separate its genetic information to make those gametes. Okay, and that original chromosome, this guy, is carrying its like carbon copy. It's holding on to it. It's like, come on, bro, you can't get lost. You got to stick with me here. And so it literally attaches itself. And each member of that homologous pair needs to do that. So you get this double X structure and they kind of travel together. Okay. All right. Questions on that part? Because when we look at recombination or crossing over, this is really important to kind of know, like, this is why you see two X's together. Okay, so in the chat, this is where you're gonna have to be like, yep, got it. Like, you need to show me that you're alive. Otherwise, I'll assume that you are not paying attention and then I'll have to like contact parents and that's not fun. Good, 2 people got. Oh, good. Yeah. Okay. Yeah. All right. Now, when you look at these, so just for funsies, this slide right here, okay? Remember I said how your chromosomes are organized, okay? We organize them into like sets or volumes, like, so we have these 23 sets of chromosomes. When they are not duplicated yet, so when they have not formed that X, they have not made a copy of themselves, so they're not ready to make sex cells, they're not ready to divide themselves down to that sperm or egg yet. They look like this. And these are actually the structures that your chromosomes make. They're not colorful like this, like I wish, but we dye them this way. And see these striping patterns that they all have? Those striping patterns represent different regions where you have different genes, okay? So we purposely, as scientists, color them so that we can, one, identify them, and we can see where you have, this is called a banding pattern. Okay, so it helps us recognize them. So we organize them into their pairs. So each one of these would be a homologous pair. So we'd say homologous pair number 12, 13, 14, 15, all the way up until you get to this last one where you don't see a number, you just see X and Y. That is your 23rd pair, and the 23rd pair, we don't really use a number, we just use the XY. That's what determines your sex. So are you born male or are you born female? If you see an XY, this representation is for a male. So XYs are males, XXs, so if there are 2 of these chromosomes together, that would be a female. Now this has a name, so when you see something like this, it's called a karyotype. So again, I'm not going to test you on that just for fun, but this is what it would look like if they were actually ready to go through that division process to make the sperms and eggs. So they've got those little belts on, so they're holding on to their copies so that they don't get lost. Okay, and this means that they are ready to go through that division phase. So if you see this, you're like, oh, that cell is going to start to divide soon. Okay, it's doing something. Here, not so much. Okay. All right, so let me go back out of that. Now, in your notebook, okay, we're going to model through what happens next, where we're going to get our first layer of genetic variation. So you're going to go into Google Classroom, and you're going to go to today's lesson, so lesson 4. for modeling sexual reproduction. Okay, and you guys will all have a copy of the Google slide. So you're each going to open up your own copy. So it starts like this. And we're going to model through this together. Okay. So what you see here is a set of homologous chromosomes. Okay, so you know that this copy right here and this copy, so these are copies of each other. This is a copy of each other, but this guy and this guy, right? They all have the same type of information at the same places. Okay, so these genes, these instructions all code for the same type of trait. This codes for the same type of trait. This one codes for the same type of trait, same and same. All right. Now, the reason why that's really important is because what happens next? So right before the separation process begins, these guys do a little barter like trading. They swap. Okay. Now, what happens is that these chromosomes literally are going to snap off a section of their leg. Okay. They're going to break off a piece of their chromosome and they're going to swap it. Okay. So think like if we were going to stand next to each other and I literally detached my leg at the knee and like broke it off. And I said, here, you want to trade me? And then you were like, yeah, sure, that sounds pretty cool. You got a good looking like calf. Okay. You took yours off and we traded and then we popped it back on. That is what the chromosomes do. Okay. So how we're going to model this is that you're going to click on this number 2. Okay. And you're going to move it all the way over and you're going to break it off. Okay. Now what happens is that now that this guy is free, you're going to click the F4, this little section, because remember, this gene is still the same. It's the same set of instructions. Now, what it, like the info inside of here might be a little bit different, but the type of trait that they're coding for is the same. So this could be like tongue rolling. It's still tongue rolling. And then this guy just gets drug right over into here, snaps back on. This one slides over. Oopsie. And replaces it. Okay. So now what you have is something that is completely different than when you started. Now this chromosome is no longer identical to its copy. Right? So this one is no longer identical to the copy that it just made. And if you're no longer identical, that's adding more variation. Okay. So, and now think about it. This was done to one set. How many sets do we have? 23. Each homologous pair can do this and does this. So when you're talking about adding like differences, let's switch things up, let's get some unique stuff going on here. The fact that these chromosomes can literally like Frankenstein themselves, like here, let me just go ahead and snap my knee off. Oh, you want to trade these? Awesome. Flip flop. Each of those 23 pairs can do that. You've just added 23 new combinations of chromosomes that are going to show up in those sperm or eggs, just right there, without doing anything else. Okay, that's called crossing over or recombination. So we are going to recombine our genes. Crossing over just means we're going to crisscross. We're going to flip flop, cross them over. And the only time that can happen is when they're in this X shape with their homologous pair. This is the only time this can happen. So it happens right at the very beginning of meiosis. They're standing there. It's like, hey, bro. Oh, hey, man. What's up? Hey, you want to trade these? And they're like, let's do it. And they flip-flop. No other time are they going to do this because no other time will they be in this state. Okay. Yeah, Brock. Yeah, your hand up.
I was gonna say something about the knee swap thing when you said it because my knees are terrible. So I would gladly do that.
Yeah. What I like to think about, like, or if you think like my friend lost her leg in a boating accident, so she has like a prosthetic, like it attaches at her knee. All I can think about is my friend Brianna going and taking off her like leg and having like her like, I mean, essentially like a nub on her knee and being like, "You want to swap these with me?" And like grabbing them with a friend, be like, "Yep, let's put another one on," and now they look different. So you're mixing and matching constantly. And every time you mix and match, boop, 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 with all 23 of your chromosomes, you're just adding to the differences, right? Because this combination did not exist before. It looked like this. It never looked like that. So this is a whole new chromosome. This one never looked like this before. It looked like that. So now we've created a whole new combination of chromosomes. And we've done it 23 times. So we've essentially created 23 brand new chromosomes, okay? Actually 46 brand new chromosomes because each one of the pairs just did this. They did it with, okay, because you had that copy there. So that's a huge amount of new variation coming in, right? Okay, so we're gonna bounce over real quick into your notebook. So slide number 16. Okay, and we're gonna go ahead and I'm gonna give you guys a chance to, we're gonna answer questions 2 and 3. Okay, so 2 and 3. So describe how your homologous chromosomes changed and how does homologous recombination or crossing over increase genetic diversity. Okay, and when we say genetic diversity, we're talking about the amount of difference, okay, the amount of variation. So take a second. I'm gonna have you guys jot down your responses here and then we'll, okay, you guys will be able to do that. So remember, when we talk about what happens during recombination, so one copy of each of the chromosomes that make up the homologous pair. So remember, I'm kind of using the word copy. So one copy of each of the pairs is going to break off a section of genes. so a section of the chromosome, and they're going to swap it with the other chromosome in the pair. When that happens, when they swap these sections of themselves, they've now created a brand new chromosome that is different from the original. Okay, and this increases the amount of variation. Okay, questions on crossing over or recombination. So here's our next like show of like sign of life. You got it? Yes, or like, ooh, no. So in the chat, yep, you're good, or no, 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 no. Awesome, Brock. Great, Bryson. Good, Gavin. Good, Mary. Okay. So the next part we're going to look at is this whole idea, okay, of what we call independent assortment, okay? So if we go back to our modeling activity, okay? So click back over into your Google slide, all right? With the homologous chromosomes you just tweaked, okay? You just made some little modifications there. Now we're ready to go through the process of meiosis, which is the process of making these sex cells so that we can actually go through sexual reproduction. Now, the process of meiosis is actually here to make these sex cells, and it's really what's happening is we just have to break apart the homologous pairs. We have to break the set up. But see how I have the original and a copy? An original and a copy that's kind of making up this set right now. This means that I have to break them apart in two different phases. Phase one is going to separate each of the pairs from each other. So it's going to separate mom's information from dad's information. Phase two then is going to actually break the sister chromatids apart, the copy from the original. Okay, so there's two different phases of meiosis here that are taking place. So during that process of making the gametes or the sex cells, we had crossing over that started up here. Now, there is another spot where you are going to see some variation that can take place, and it happens right in here, and it's called independent assortment, okay? Now, an independent assortment, okay, this means that the chromosomes, okay, these guys, the way that they're going to split, there's a huge amount of variation in how they could do this, because what they have to do, if you're going to go through like a process of moving, it's going to be very orderly in how they do this, okay? So I'm going to bounce over to my dot cam really quick and kind of walk you through what this is looking like. And then we're going to model it out because it's kind of hard to see when we model it, really kind of what's taking place. So basically, okay, what you've got here, let's say we had those two sets of chromosomes. So you had your first set, okay, and you have your second set. All right, and I really should say, like, remember that guy's like that, and that guy's like that. Okay, so we've done our first little like switcheroos here. Now, remember, part of this is we have to separate these guys from each other during this first phase that we're going to go through. Now, when this happens, and I'm going to actually say that we're going to have two sets of chromosomes. So for this one, just to make it a little bit easier to show you guys. So we'll say we have a big set of chromosomes and we have a little set as well. So we have a big pair and a little pair. Okay. Now, there's a lot of different ways that these guys can line up to be separated, because when we separate, we literally are just pulling them apart. So your cell just goes Yep, nope. We're going to cut you right down the middle, and you guys are going to go this way, and you guys are going to go that way, right? Now, who's to say? So this is one way they could have lined up, okay? Which means that now these pink ones are going to go over here, and these blue ones are going to go over there. But these guys could have lined up like this, okay? Just as easily as they could have done this, all right? Now, if the cell now was going to split in half, and these guys went this way, and these guys went this way, that creates two different options. So this would be like option one. This would be option two. Now it gets even wackier because these guys are not connected. The little set and the big set, they're independent of each other. So this one could have been here, but this little set could have been like, no, we're going to do this. So now what you get is that when these guys separate, those two go this way, those two go that way. So notice how this pair had both pinks this way. This one has a pink and a blue. This one has a blue and a pink. So when I go to further break those down, the sex cells that this assortment or this arrangement's going to make are going to be different than the ones that this arrangement's going to make. Same thing over here. You could have let's say you want the blue on this side and over here, and you want the pink right here and the pink over here. And then they're going to split and they're going to pull those guys this way and those guys that way. Okay, this is what we call independent assortment. The random lineup of how these chromosomes are going to arrange themselves before they get split up. That's independent assortment. Now, I did it with 2 chromosomes, okay, 2 pairs. How many do we have, though? Now, if you have 23 X's and they can all arrange themselves, so let's say this one wants to go that way, then you got the other one that's going to go this way and that way and this way and that way and this way and that way, and you've got 23 different chromosomes with all these different options to choose from. Think about all of the possibilities that you're going to end up with in these sperm and eggs at the end of meiosis, which can actually be calculated. Okay, so questions on this part right here. So during that process, when we are splitting these pairs, the big X's apart from each other, the way that they're going to line up, okay, because they line up kind of like in order, like when we literally say line up, we're going to move them. They have to stack on top of each other, and we're going to split them apart. And the way that they line up could be different. It's random. We call it random assortment. Okay, random organization. Okay, and in this means that we end up with just insane amounts of possibility. Okay, so much so, do you guys have any idea how many you would end up with? Okay, if all of these chromosomes had to line up and separate? Care to take a guess in the chat? How many different combinations? Okay, see how close you guys can get. So how many possible egg or sperm combinations do you think you could come up with if we had to line them up and separate them? How many different types of sperm and egg could you come up with? So in the chat.
Probably hundreds of thousands.
Do you think like hundreds of thousands?
Or thousands.
Now remember, you have 23 sets, so you're going to have like not 2 in line, like I showed you 2, just because who wants to draw 23? So you have to apply what I did to 23 pairs of these things. So 23 partners standing in line, but they could be left and right or right and left. Like,
okay. Is it 23 squared?
Close. I think you're on the right track.
Yes.
46 squared.
Okay. It is, let me see what I got. 8,388,608. Oh, Carson, you were close. That's not 23 squared. It's 2 to the 23rd. Okay, so when we talk about genetic variation and possible like options, there are 8,388,608 different possible arrangements that you could come up with for these guys to break apart into different sperm and eggs. So when we talk about like, oh, you're 1 in a million, no, not even close. You're 1 in well over that. Millions.
And we're
Okay?
not
And
even
I'll
at the
blow your
last
mind here
stage
a
yet.
little bit. What was that?
And we're not even at the last stage yet.
No, because we haven't even added the next layer of variation. Okay. So if we go back to our modeling activity, okay, to kind of show you again, like so you can like really kind of feel what's happening here. What you're going to do is you're going to click on one side of your chromosome, and then you're going to hold the control button down and click on the other partner. So that way it grabs both of them, and then you can let go of the control. And then when you have that little arrow, you're going to be able to then pull him that way. That simulates that first level of breakdown. So I would click, and you're going to do it to the next one. Click, hit control before you click the next one. Click it again, so that way it groups them together. And then you're going to drag. All right. Now, those are the two major things that take place during the process of meiosis that adds variation, okay? So it happens right before we start and right here when we're splitting apart our big X's, our homologous pairs. Now what we need to do is we need to separate its copy from itself. So we basically do the same thing. They're going to do a similar process where they're going to line up, but instead of splitting the big X's apart, we really are just pulling each part of that X into its own individual gamete or sperm or egg. Okay. Now remember that original cell we started with had a complete set, one copy from mom, one copy from dad. But in the process of meiosis, we are breaking apart those homologous pairs so that we can just donate half of that because we know during our next stage of creating an offspring, fertilization, that other half is going to be joined together. So in each one of these, so in this egg, you're going to have one copy of the pair from mom, but then notice this one. This was the other guy, but it's different. This was the copy from dad, but then that's different. So not only do we have, 8,388,608 different possible combinations of how these are all going to look if we had all of our chromosomes here, but then each chromosome, because of crossing over, is different. Because they got their legs ripped off, yes. And they are like Frankensteining each other. Okay, for sure. Questions on that part? Because we're going to go to our notebook and fill out that page, that next section. Okay. So now when we go to our notebook for modeling independent assortment, and that term independent just means that it happens without any influence. These chromosomes line up how they dang well choose. Okay. They're not influenced in any way. They respond independently and they do it independently of each other. That's where the variation comes in. So just because set number 1 wants to line up mom, dad, set number 2 is like, what if I'm going to go dad, mom. And then the next one could be like, I like dad, mom too. And then 4th could go, no, I like mom, dad. Meaning like how they're going to set up themselves. Okay. So when you're looking at question number 5, you guys take a second and you guys are going to describe how your eggs are similar and how they are different. So how are they similar and how are they different? I forgot, also do number 6. Okay. How you guys doing? How are you guys feeling about independent assortment? Okay, that second one. So yes is in the chat if you're like, yep, I got it. No's if you want me to kind of walk you through it again or in a different way. No, is that a meh as in like kind of get it, or a meh as in like you're like an in between? Okay. The biggest thing I can kind of tell you is like, let's say that we were at school, okay, and we were really going to separate you guys into groups, okay, and we partnered you up and you guys all had to get in line and you were going to be in line with your partner. And so you created this like single file line, except you weren't single file, you were with somebody. You were in twos, but you created a line, right? Okay, so when we're showing the big pink X's and the small ones are able to, yes. So now let me so in this example, so let's say we had 46 people and I said, okay, partner up, right? And you all like found a buddy and you're like, okay, I got my partner. And then I said, okay, now with your partner, I need you guys with your partner to form a single file line. So really you had a double single file line right next to each other, right? Okay. And I said, now arrange yourselves in as many different possible combinations as you possibly could. But you have to stay in this arrangement where you have 2 people, 2 people, 2 people, 2 people, 2 people. Okay. That's basically what independent assortment is. So when the cells are going to get ready to separate, they're going to essentially, when you say separate, we're talking about movement. We're moving genetic information. Now, think about when you were a little kid. When you moved from one classroom to another when you were in elementary school, what did they make you do? Did they let you just run willy-nilly down the hallway to where you were going to go? Or in most cases, they made you walk in a single file line, right? Let's all walk together. Let's walk in a single file line. And that's what you're going to do. However, the way they have a lot of freedom in how they're going to like arrange themselves, okay, is partner number one going to be on the left or the right? Okay, they could be on either. Okay, and each time they go through this, they're not held to their spot. They could flip flop. So, like Mary, if you and I were partners, you'd be like, okay, I want to be on the left. I'd be like, all right, I'll be on the right. But then the next time we went through this, you'd say, oh, I want to switch to the right. And be like, okay, I'll be on the left. So just right there in our ability to flip flop, like, are we on the left or are we on the right, is going to create a brand new sperm or egg, brand new option, because each partnership, each of the 23 pairs that are in line are doing the same thing that we're doing. So you're coming up with all of these different possible combinations, because once we get to like our destination, essentially, if I was a teacher, I'd be like, okay, now group, partners, separate from your partners. And those are the teams. Each team is going to be different each time because we all lined up differently. Does that kind of make sense? So when you are separating and you're making these bigger groups, that's kind of what's happening right here. And when you're selecting, it's like the way that they're lining up together is completely random, okay? And it changes all the time. So when we talk about those possible variations, that's why we say there's over 8 million, I think 8,388,608 different possible ways for those chromosomes to line up before they get separated. So that means that there's 8,388,608 different possible combinations for these sperm and egg, just in how the randomness of how they're going to line up together. Okay, but they can only flip flop with their partner. They can't, like a big then can't be with a little. So it has to be like you have to stay with your buddy. Okay, you can just decide if you're going to be on the left or the right. Now that's one layer. Okay, we have this next layer, and it has to do with when we get to fertilization, the like crux of sexual reproduction. So when we are going to start to combine the sex cells. So when you have a sperm that's going to come together with an egg. Okay. Now, when you have a sperm that comes together with an egg to form that genetically different cell, we call that new cell a zygote. Okay, so when we were all first like came into this world, you were a zygote. Okay, that was the first phase of your existence, was a zygote. Okay, now when you have a sperm, this is specifically in humans right here. Okay, notice how this guy has 3 chromosomes and this egg has 3 chromosomes. Okay, each one of them is going to make up 1/2 of their set. Okay, and when they combine together, all of a sudden they're reunited. Okay, not a goat. You just pronounce it goat, goofball. Okay, now once you reunite these guys, their homologous pairs are reunited. Okay, and now we're back to having a complete set. Because remember, the way that our initial information, the way that we have to have our stuff, we need it in sets. Okay, so you need a pair from mom, a pair from dad. This is a complete set of all of your genetic information. When it's in the form of a sperm and an egg, you are missing half. So when those two things combine together, we're reestablishing those sets. Yeah, and here's how it gets even better, Mary. So when someone says, oh, you're 1 in a million, it gets better. So how many possible zygote combinations do you think there are? So when we talk about what possible variations we can come up with, how many possible zygotes combinations are there in humans?
I think that just takes
what was that?
I think that just takes the 8 million chance we had before and multiplies it times 2. Because we have the 8 million from the mom and the 8 million from the dad.
No, not quite. So anybody you guys want to guess?
I just wanted to try to say 8,388,608. I just wanted to say that.
Okay, drum roll. You are not 1 in a million. You legit are 1 in 70,368,744,177,664. Yeah. So when we talk about genetic variation, we're not messing around here. Like, this is straight up like, what? Now, think about how many people there are to reproduce with. This is just based off of when you combine the variation between one sperm and one egg, because that number is based off of 2 to the 46th power. That is not taking into consideration of, now we're going to add variation in terms of what we call like sexual selection. So like selecting a partner, which is now you have to take into the population of all humans, like reproductive candidates. Who could you reproduce with? So this stuff is like no joke. When we talk about we're adding genetic variation, oh, heck yeah. We are all like, that's just crazy though, because when you think about what we talked about last unit, we're essentially like the same, we have the same genetic code for nearly all of it, right? We're only 1% of us is really kind of different in terms of like the types of things that we have, but we have all this built-in variation within that like 1%. Yeah.
You also got to realize all the people that have been alive or have just died and stuff. So overall, all the people that have been on the world, there's been genetic variation to all of those.
Yeah, and so then when we think about, when we get into our next unit, we're gonna, and kind of where this is going is, so what's the chances of getting certain traits to come up together? Or what's the chances of inheriting something? What's the chances of a particular genetic disorder building frequency or building numbers within a population? What influences that? So then how do we see things kind of stick around over time, or why do we see that? Those are kind of the next two concepts that we're going to be covering. Okay. Yeah, way too big a number. All right. So when we talk about like these things build genetic variations, we're not joking here. Like, we're talking big time variation.
So how did we get the number, the 70,368,744,177,664?
I know. Who wants it? Wouldn't you want to have that like in your bank account right now? Like, yeah, Carson, you have your hand up.
How did you get it though?
Oh, 2 to the 46th power.
I wasn't really listening earlier, but let's just say we have a bunch of eggs that are genetically identical and a bunch of sperms that are all genetically identical. Will they all produce the exact same baby?
Essentially, if none of these because if they're all identical, then they didn't go through crossing over and they didn't go through independent assortment. If they were all
as in like they went through that, but like each egg did go through crossing over, but each egg is identical, like to each other.
But because of crossing over, they are not identical to each other.
Yeah.
So if you go to like this guy.
But what I'm saying is in fertilization, it's not like the genes from the egg and the genes from the sperm combine in a way that makes them unique. It's just that the fact that they're combining is what makes it genetically unique. Because there's so many sperm and egg that could.
Yeah, so this combination, because this is coming from a different gene line. So the sperm already is coming from a different source. So the type of information and that's coded for each of these traits is going to be unique from this egg already. So when you are looking at this, but each egg that could possibly be fertilized, right? Or each sperm that is like could possibly fertilize this egg, each one of those sperms is different. So it's like random which sperm is going to actually be the winner and to be able to fertilize this egg to become a zygote, to combine the DNA as well.
Honestly, I'm not even sure what question I was asking. I really confused myself.
I was kind of confused. I'm like, so the big thing here is that knowing that the crazy part about it is that every time your body creates its sex cells, each one of the sex cells is unique to the other. So in females, each egg that's produced is unique.
But what if we have future technology so we can clone the egg cell so it wasn't? Like, we get one egg cell that is unique, and then copy that egg cell a bunch, and then do the exact same thing with the sperm, and then all those combine, and would each baby be unique?
Well, eventually each baby would then be just a copy of itself, right? Because you're using, but basically if you were going to do that though, aren't you doing asexual reproduction? If you're just carbon copying all of the gametes, like all of the eggs were genetically identical, all of the sperm were genetically identical,
and that would
then all the offspring would essentially be like identical.
Okay,
they would
that
all then
answers
have
my
the same
question.
combination, right? So that right there, you would have to think about like what would be the benefit of that. We would be losing a lot of the benefits of sexual reproduction because we'd be taking away a lot of this genetic variation that's built in.
We'd become technically a virus, and we could, or bacteria, and we'd be able to be killed off more easily.
Yeah, when you stick to like limiting a lot of the variation, like a lot of this from happening, then yeah, you are essentially kind of chipping away at our like invisible armor a little bit. Okay, keeping us alive. And from a like species standpoint, remember, like human population, like not just from an individual standpoint, we're talking very like for the whole human race. This is what this applies to. Like when we talk about increasing our genetic variation, we're talking about as humans all together, not just one person to one person, right? So this is very large scale thinking right now. Okay. But the last part of if we go back to your modeling reproduction so that we can finish that up and you guys can submit that, okay? You guys are going to get to make a zygote, okay? So basically what you guys get to do is that you guys are on the right, you have all the possible eggs from our little example here and all the possible sperm, okay? So for simplicity, we just kind of modeled it for what it would look like for sperm. You guys made eggs. We just did the same thing for sperm, okay? But when the sperm actually comes together and penetrates the egg, okay, this is what you guys are modeling, and so that's what forms that zygote. So you get to randomly select whichever ones you want to select, okay, to move in there together to create a homologous pair, so a complete set of genetic information. So a zygote that contains a copy of genes from mom and a copy of genes from dad. Okay. So I could go, whoopsie. Careful when you grab at them because see what I just did there. So you have to make sure that like when you click in it, you're only selecting the actual chromosome. So I can move that guy down, whoopsie. And you might accidentally like pull its little Frankie leg off. Let's say I chose those two, okay? So now what you guys have here is that zygote with that full set. Okay. So for this one, this is where I'm going to let you guys kind of figure it out. All right. So you guys get to figure out how many different zygotes could you possibly make. Okay. So what are your combinations? You guys can try to see if you can do the math, or you can literally drag and drop each one. Okay. And then using that info, you guys are going to finish up and you're going to complete the last 2 questions on slide 16. Questions on fertilization. All right, guys, so that's what I've got for you today. So with the next like 15 minutes or so, okay, I'm going to give you this next little bit of time. Thanks for hanging in there for the entire time. I know that was kind of like a long one, but we did have a lot of notes. I wanted to make sure you guys had that stuff down. Okay, so you want to make sure that you complete the Google slide and then you hit submit on Google Classroom. Okay, and then you finish up those last 2 questions on slide 16. Once you feel confident, like you're like, okay, I could explain each one of those, you're gonna then at some point today, so this could be your homework, you're gonna jump in and take part 2 of your quiz. It's 5 questions, okay? So think of it like a big exit ticket, all right? And then at that point, I'll have both of your quiz scores and then I'll add them over. If you have not done part 1 yet, add that to your list. So take both if you haven't done both, or just part 2 if you already did part 1. And then last kind of thing, if you still needed to finish up anything from slide number 13, now that we've gone through each of the parts or all of that information, go ahead and do that, okay? Because then tomorrow we're going to be jumping over and starting to look at adding some vocab for how we figure out what are the possibilities of different combinations showing up together, okay? All right, so use the rest of this time to work. You guys can stick around. I can put you in breakout groups. You can stick around and ask questions, or you guys are free to jump on out and kind of wrap up those final pieces, okay? Other than that, have a phenom day. Remember that you are not 1 in a million, but you are 1 in over 70 total alien, which makes you super awesome. So have a super awesome day.